To you all, I designed this type of sampling because I was doing a lot of backpacking and literally carrying a bag, for 10-day field trips in the backcountry. And so I wanted something super lightweight, something that you could actually just go in with one unit of this and bleach it. So a unit for the sampling is basically a pair of gloves, a bottle, a syringe, and a little filter. And that's it, that's all you need. So it's really fast. You don't need this hand pump thing. Our syringe, you push it through instead of suck it through. So it's just a different idea. The other thing about this method is if any of you have done water quality assessments, where you sample water, you usually have to filter it in the field, and then you take that water and take it back to the lab and analyze it for the nitrogen and phosphorus species and things like that. So this method, and actually the filter that they use for that, is exactly the same for this method. So it's a glass fiber filter that's typically used for that method. And what you can then do is, if you have the ability to keep that water sample, you can get water quality information at the same time as the DNA. So the water quality people just threw that filter away and they didn't know what they had. So so that's why this method evolved as it did. And so I'm gonna demonstrate one, and then I have enough for about 12 of you, and these aren't real samples, so they don't need to remain independent. So if anybody wants to try pushing it through, we can. And just so you get a feel for how to do this, I will show you the basic sterile technique as well. And anybody who wants to do that can. So the most important thing about this is not getting human DNA in the samples. So for target species, it's not a problem because you're going after DNA that's not going to match your own. But when we go to these metabarcode approaches, we amplify our own DNA because we're eukaryotes, we're animals. And so the most important thing, if you take a sample that you may want to use for the universal sort of metabarcode approach, you want to make sure that you minimize human DNA contamination. So the gloves are part of that. What it does is you waste your potential to detect the other species by sequencing a bunch of human DNA. And so you're going to get, 5 million sequences, and if half are human, we know humans are there. We don't care about the humans, right? So it just wastes the potential of what you could do. That's the problem. So it's, yeah. Okay, so gloves. Then, if you set this up in its own unique bag, you could have one filter housing with the filter already preloaded inside there, so you don't even need to handle it or do anything. And then you have a sterile syringe. Open it this way. There we go. Okay, there you go. So you have your sterile syringe. Of course, if you're in I always have an extra bag if you're like in a windy area so that your trash doesn't blow away. You can create a little trash bag. So the syringe is a lure lock syringe. If you haven't ever seen these, they just screw on to the top like so. And now your filter's attached to the syringe. And so that's ready for that. And I'm gonna put it in here. So to take a water sample, we just have a 250 milliliter water bottle. So if that's the goal, 250 milliliters of water, you can create your sampling vessel to be that size. You can also, of course, take a bigger sample. It just depends on if you need things to remain independent or not. So you could try to keep it closed until you're to the surface of the water. And then you're just going to dip it down and fill it up. And then if you were moving, going multiple places, if you were going to sample multiple sites, you would change your gloves between each one, right? So the idea is that you could just sample one sample and then filter it, change your gloves, and do the next one. Those are the types of things you'll have to think about when you're out doing this, okay? And then You have to have this unscrewed to be able to suck the water in. So these are wide mouths, so you can fit it in. One of the things you don't want to do is push it too far in, because then all the water squirts out. So you just suck up some water. You try to go to the 50 ml mark, and if you want 250, then you do this 5 times. Okay, and you can squirt it back on, and then I'm just going to squirt it back in here, but if you were doing a system, you could just put it on the ground somewhere. It doesn't really matter where. You don't want to contaminate your stuff, so then you just push it through. And in the beginning I was really sensitive about how fast and how hard I was pushing and all that. I don't think it matters. So just push your water through. Okay. And then you would do that until you have the desired volume of water that you can fit through. Or once it becomes too hard to push, then you might have to stop and change the filter. If you need to do a certain volume, if that's your target, then you might have multiple filters and you just combine them later. So that's no problem. One of the big things that happens is when you push the water through, there's still residual water inside here. So it's really, really important to make sure that you blow air through it to get all that residual water through. And that's because of what we're going to do with the filter next. We're going to store it, and we want to just try to get it as dry as possible. So you fill your syringe with some air, and then just push all that remaining water out as best you can. There you go. And that usually does the trick. Sometimes you do it once or twice, right? Okay.
Do that each time?
No, just when you're done. Just when you're done with the amount that you want. So then, usually you have some kind of thing you've set up, like something like this, where you could process this. And you just have some tissue that you can decontaminate your surface with. So just some water, or some wipes, sorry. And then there's this handy stuff called DNA Away, which is basically glorified bleach. So you can pay a lot of money for this, or you can just take your household bleach and dilute it to 10%. I just brought this so you could see that you can do this. So you could basically wad up your paper towel, clean off your surface to make sure you don't have any residual DNA, or if you use it for multiple sites. Okay, and then have your little trash bag handy. Okay. And if you feel like you touched a lot of stuff or anything, just change your gloves, especially for this phase. So what we're going to do now is we're going to take the filter out and we're going to preserve it. And so if you feel like you just touched a bunch of things or whatever, just change your gloves. If at any time that feels like a problem. And so we have something, we just have a bunch of tubes, and inside a tube is basically a buffer. So Kath talked about having the filter, you dry it or you put it in ethanol. And what we've optimized is basically this is the first step of the extraction to extract the DNA. And we've done some tests where it can actually sit in this buffer for up to 3 months and not have a negative impact. And so if you're out in the field and you're backpacking, it can be sort of not at extreme temperatures, but sort of, a good range of normal temperatures, not like 40 degrees and stuff in direct sunlight. But we haven't tested those conditions anyway. But you can see we're going to put the filter directly in here. Okay. We have these disposable tweezers. And like I said, if you were doing this in a situation where you don't need very much stuff, you would have this, your box, maybe this, and a pair of tweezers, and you could do backpacking with that and then sterilize in between. So that's what I was trying to do, minimalist situation. Okay, so now our filter has been water put through it, we dried it, and now we're going to put it inside this tube. So this just unscrews. And if it's windy, you have to be really careful. So sometimes it's good to get low to the ground especially the area where you've decontaminated, because if it flies off, you have the ability to catch it. So if you're down pretty low, it should stick to there pretty well. If not, if you're okay with your gloves being clean, then you can kind of stick your finger on it to hold it in place. And then you're just gonna fold it up like so. So all this stuff's on the front part. So once you fold it, you can put your thumb on it, fold it again, then you have it in your tweezer, and you put it inside the tube. And then you can take one side of the tweezers, push it down, make sure it's underneath that buffer so that it's getting preserved. And you close it, and that's it. Of course, label it and write it down on your sheet. And then you can move on to the next one. So if you bring enough stuff, then you can have each one of these already set up independent. If you want to reuse stuff, then maybe you have a little vessel that you can put bleach in and decontaminate things as you're working.